Hello, everyone, and welcome to our 8th lecture in Psychology 1000. In this lecture, we'll begin a new unit, and this unit is about the senses. So, in this particular lecture, we'll talk about sensation, perception, and we'll talk about the visual system, which is the sense we rely most on as human beings. So, by way of overview, uh, we're talking about the senses, so that is how we get information from the environment, from the world around us. So we'll start by talking about sensory systems in general. What are their properties? What do they do? And this is something that will apply to all the senses. Then we'll start talking about vision in particular. And so the first step in vision is receiving light. And as we'll see, the visual system turns that light into some sort of neural signal. And then, of course, once the light is taken in by the senses, by the eyes, it has to be processed. And eventually we end up with a sense of the total visual scene. So our brain has to do a lot of processing to understand what's going on in a complex environment like the one we usually inhabit. Our world doesn't exist in terms of simple lines and contours and simple colors. It's more complicated than that. And so what we're going to do in this session is talk about the difference between sensing, simply receiving information from the environment, and perceiving, creating a mental representation of what's out there. We'll also understand why this is a hard problem. So sensing the environment is not simple, and even though our senses do it in a way that we take for granted. So we don't think about a lot of what our senses tell us. We sort of interpret it automatically, and a lot of that's unconscious. But a lot of processing goes into that. For vision in particular, we'll see how the eyes get information and send that information to the brain with a bit of processing. Uh, but of course, it's also the brain that takes that information and processes it even more. So the brain processes the information, takes it and turns it into mental representations. We don't see simple lines and contours and dark shapes and light shapes. We see complex objects, familiar objects, new objects, objects that may overlap, that may be at different distances. How do we interpret that visual scene? So we'll talk about how the brain does that. Up front, some terminology. So we have a variety of concepts to deal with. We have, for example, the object itself out in the environment. Uh, and of course, we don't have pure access to what that object is. It's filtered through our senses and through our perception. So we have the object as it is out in the environment. We have our sensory system. In this case, I'll use the eye. We have the brain, our information processing organ. And then we have more abstract concepts like mental representations or idea of what the stimulus looks like. So in this slide, I've used a hot air balloon. And our mental representation of what's out there, of that hot air balloon, may not perfectly match what actually exists out in the world. And that's just something we have to live with. Our mental representations are not perfect facsimiles of what's really out there. So some terms. So first there's sensation. And we talk about colloquially what a sensation is. We have a sense of something. Uh, but literally what a sensation is the stimulation of a sense organ. So in this case, the eye, or more specifically, the retina. So when that piece of tissue becomes active, it's because there's something out there in the environment shining light into the eye, for example, or reflecting light into the eye. And this is true for other senses as well. Pressure on the skin, for example, or sound waves being transmitted to our sense organs in the ear. We'll talk about that next unit. But sensation is just literally that. It's the stimulation of the sense organ. Nothing's gotten to the brain yet. We are not aware of the sensation. We're not aware of the stimuli out in the environment. The sense organs have just been affected by it. So that's this first step here, getting from the actual stimulus out in the environment into the sense organ and activating it in some way. Then there's transduction. And that is the way the nervous system converts the stimulus into neural signals. So in the case of the eye, we get from light into action potentials. In the case of sound, we'll get from sound waves into action potentials. In the case of touch, we would get from pressure on the skin into action potentials. So all these things have to get converted into neural signals. And these are, out in the world, are different forms of energy. So we've evolved these different sense organs to detect these different kinds of energy and convert them into these neural signals. So that's the second step here, getting from the eye to the brain, turning the stimulus from light into action potentials that the brain can then process and interpret. And then the last step is what we call perception, and that is the interpretation of the sensation. It's the interpretation of the neural data that are coming from the sense organs. So the brain is just sitting inside your skull. It doesn't have its own sense organs. And so it relies on information from sense organs to process and then to interpret in order to figure out what's out there in the environment. And so the brain's job, in terms of perception, is to turn those neural signals into a mental representation, the idea of a hot air balloon in this case. And so that is what perception is. So sensation and perception are very different things. Sensation is in many ways the first step. Perception is the last one, getting a mental representation. Something has to activate that idea of the stimulus or the object. And so those are terms that are important to differentiate between. So of course we didn't always have this conception of the senses. And we didn't always know how to approach this problem, how to get from objects in the environment, stimuli, to mental representations. And so one of the first scientific efforts at this was the field of psychophysics. And what psychophysics does, and it's still a field that's very much alive, what it does is measure the relationship between stimuli and what we call the observer sensitivity. That is the observer's ability to discern stimuli. So that is sort of the first, and to a small degree, the second step in our scheme of getting from environmental stimuli to mental representations. So for example, we have the concept of the absolute threshold. And this is something that can be measured. And that's one of the points of psychophysics, is it's not really looking at abstract concept or mental representations. It's looking at people's ability to detect stimuli in the environment. And so the absolute threshold is the minimum intensity that a person is able to detect half the time. If they're not able to detect it at all, then obviously it's below that person's threshold. If they're able to detect it every time, well, that's a pretty strong stimulus. And so the criterion here is, what can a person detect accurately 50% of the time? So for different senses, we have different thresholds. For example, for vision, there's evidence that we can detect a single photon, a single light particle. And this is represented by, for example, a candle 30 miles away. And your book makes the point that as long as it's a clear, dark night, it's not obscuring the light, it's not diminishing it. But a candle 30 miles away is a pretty subtle stimulus. And yet, for a lot of people, that is their absolute threshold. It's not for everyone. Some people may need more light than that. For hearing, you can, for example, have a clock ticking. And if that clock is 20 feet away, that ticking sound is very soft. But for a lot of people, that is what they can detect about half the time. That's the minimum intensity for sound. For touch, a fly's wing falling on the cheek. Of course, that's an odd stimulus to pick, but that is an example of an absolute threshold, the light touch of a fly wing falling on the cheek. And of course, depending on which part of your body you're talking about, your sense of touch is more or less sensitive. Smell. So, example, a single drop of perfume in a large volume, so 6 rooms worth. That's a little bit of chemical that your senses can detect. Your sense of smell can pick up those small amounts of those chemicals that are in the perfume. Of course, other animals have a much better sense of smell than we do. Ours is really not that good. And taste. Our sense of taste is not very acute either, but you can detect a teaspoon of sugar dissolved in about 2 gallons. Again, this is the average person. The average person can detect this half the time. There's also the concept of the just noticeable difference, or J&D. And this is not the smallest stimulus, but rather the smallest change in stimulus intensity that a person can detect. So if you are getting some amount of light or some sound, if you bump up the light intensity or change the light color, if you make the sound a little bit louder or you change the pitch, um, if the person notices that, then that is the just noticeable difference. It's the smallest amount that they'll say, yes, that's different. And the thing is, there's a really interesting phenomenon called often called Weber's law. Weber was actually German, so more accurately it'd be Weber's law, and it was made more famous by an individual named Fechner. So it's sometimes called the Weber-Fechner law. They're all the same thing. And what this law says is that the just noticeable difference is proportional to the intensity of the stimulus. So what does that mean? Well, it means that if you're holding something that weighs an ounce, and I add an ounce to it, uh, that's easy to detect. I just doubled the amount of stimulus you're getting from 1 ounce to 2. If you're holding something that's 20 pounds and I add an ounce to it, you're probably not going to detect that because the initial stimulus intensity is 20 pounds. It's a big stimulus. And I only increased it by an ounce, not very much at all. However, if I double it to 40 pounds, you're as likely to notice the switch from 20 to 40 pounds as you are from 1 ounce to 2. So it's not the absolute amount added that's important, it's the relative proportion. So again, going from 1 ounce to 2, I double the stimulus, and that will be as intense a change, as noticeable a change, as going from 20 pounds to 40 pounds. You can come up with other examples as well for light or for sound, but that's the point, is that it's the relative change in intensity that matters, not the absolute change. Okay, so when it comes to the senses and psychophysics, uh, a lot of psychophysics is about what's called signal detection theory. And that sounds fancy, but what it really means is, uh, well, whether you're an instrument or a person, how well are you able to correctly detect what's going on out in the environment? So for example, if you have a simple stimulus, the stimulus is either there or it's not. And so you have this binary distinction. Is the stimulus on or is it off? And of course, then you have the observer, or if it's an instrument, the detector. And the observer can either say, yes, it's there, or no, the stimulus is not there. So the stimulus is either there or not, and the observer either says it's there or not. And so there are terms for all the possible combinations of those two factors. So for example, if it's there and the observer says it's there, that's what's called a hit. So you've correctly detected that the stimulus is out there. If it's not there and the observer says it's not there, that's also correct. So you correctly said that the stimulus is absent. That's called a correct rejection. If the stimulus is there and the observer says it's not, well, they've made a mistake, and that's called a miss. You've missed the stimulus. It's present. You said it wasn't. You missed it. And then, of course, you can be wrong in the opposite way. If the stimulus is not there and you say it is, that's a false alarm. And these are terms used in signal detection theory and in psychology to describe the relationship between the stimulus and what the observer says, what they perceive. And so a false alarm is not if you've missed something. It's if it's actually absent, but you say that it's there. So you've detected something that is actually not out there in the environment. So these terms come up over and over again in psychology. Hit, miss, false alarm, correct rejection. And these are what these terms refer to, the relationship between a stimulus and our perception. And of course, our responses in this context depend on our sensitivity, to be sure. So if you have really good sensory instruments, you've got a better chance at detecting whether the stimulus is there or not. But a big part of signal detection theory is also about decision criteria. And that's not something we think about very much. So if you're in an experiment and you're waiting for a very dim flash of light, if the person says, it's okay to be wrong, don't worry about it, just do your best, then you're going to have one decision criterion. If the experimenter says, if you say it's there and it's not, we're going to forego your entire payment for the experiment. So there you've introduced a real cost to being wrong, a real cost to those false alarms. And so your decision criterion is going to be a lot stricter. You're going to try and avoid those false alarms if they're going to penalize you for them. And so signal detection theory is not just about the accuracy of our senses, it's also about decision making. What threshold do we set before we'll actually say that we saw something? Because we know our senses aren't entirely reliable. So the incentives that were offered, the experimental setup, will bias our perception one way or the other. So it's an important thing to think about that often gets glossed over. We often treat our senses as though they're totally accurate, when in fact, maybe they are accurate, but our decision-making process is also involved when it's time to describe the environment, when it's time to register whether something is out there. Signal detection theory has a number of applications in a wide variety of aspects of our lives. Certainly, instruments are part of it, so when we're designing a machine, we have to set its decision criteria. So it's often as sensitive as we can make it. But for example, a metal detector may be fairly sensitive to the presence of metal, but then you have to decide what the criterion is. How many misses or false alarms are okay? So where do you set the criterion of that instrument, of that metal detector, where it might miss small metal objects? But you also don't want it going off for every person that walks through. Uh, and so there's that sensitivity. You're not going to be perfectly accurate. The question is whether you allow things that are there to be missed, or whether you have false alarms, whether the instrument says it's there when it's not. And so that's a big issue with instrumentation and deciding how to set instruments so that they're sensitive, but also minimize misses or false alarms depending on the application. Certainly medical testing, false alarms are a big issue. If you have a rare disease, for example, that you're testing for, you're probably going to have a lot of false alarms. You're going to have a lot of people that you say have the disease when in fact they don't. This is a big issue in things like mammograms. Uh, Given the incidence of breast cancer, you can have a lot of false alarms partially because the instruments are not that sensitive. It's hard to tell sometimes, but also because you have to decide where that threshold lies. Do we want to miss cancers that are there, or do we want to say that a few people have cancer when they don't? Um, so signal detection is a big issue in a lot of medical testing, even in our justice system. So our justice system is designed to be biased toward the innocent. So our criterion is not, is it more likely than not that they did the crime in the case of a criminal trial. The criterion is beyond a reasonable doubt. Well, that's a much higher threshold than probably, yeah. So the issue there is you're going to have some misses. You're going to let a few guilty people go, but that's at the expense, or that's the trade-off we have for not sending an innocent person to jail. Our threshold has to be pretty high. We have high criteria for convicting a person because we value not putting innocent people in jail over letting guilty people go free. So we have to set a criterion somewhere, and that's the issue at the heart of the justice system, at least the trial process. So this comes up in a lot of different parts of our lives. And then finally, we have the issue of sensory adaptation. And what this term means is that our senses are not equally sensitive over long periods of time. That they actually decline over time. Certainly, as we age, our senses tend to decline. But even if we're exposed to the same stimulus for a long period of time, uh, we tend to not pay attention to it. We tend to not be aware of it after a while. So we get used to odors in a room. So for example, wherever we happen to live, we've become insensitive to how that living space smells. Whereas a guest who walks into that living space, for them, the odors will be very noticeable. Your book also has visual illusions. If you stare at the same visual stimulus for long enough, your sensory system will adapt. You'll get used to that sensory stimulus being there, and you'll stop responding to it. It's the same way that we can also start to ignore repeated sounds in a room. You'll be less sensitive to them over time as you hear them over and over again. It's how we're able to tune out music, for example, or repeated noises that are distracting. We can eventually tune them out to a better degree because our senses get used to them. And the reason we do that is so that A, we can pay attention to more relevant things in the environment. We're not paying attention to the same thing that's always there, but also so that we're not wasting a bunch of energy processing a sensory stimulus. If it keeps on being there over and over again, we don't want to treat it the same way that we treat a new stimulus that actually needs to be processed. Okay, so that's all true for the senses in general. And now we're going to start talking about vision in particular. And what vision does is convert light into, eventually, mental representations of objects and perceptions. So the first question, of course, is what is light? Well, technically, a light is electromagnetic radiation. And that's a term from physics we don't need to really worry about a whole lot. But, of course, for us, light has to be visible. And so electromagnetic radiation covers everything from radio waves to X-rays and gamma rays, a lot of things that we can't see. And so when we talk about light in psychology, usually we mean visible light. But it is a form of energy, electromagnetic radiation. And it has some properties that we need to think about when it comes to visual properties of the environment. One of them is wavelength. So light is a wave, and that means it oscillates. And it has a wavelength, which is the distance between peaks. So this light can be sort of visualized as going up and down. Obviously, we don't see light that way, but if you measure it, it is a wave. It has peaks and troughs. And the distance between the peaks is wavelength. And wavelength of light is what, if it's in the visible spectrum, what we perceive as color. So for example, red colors have a longer wavelength than blue colors do. So it's all light, but the wavelength of the beam of light, of the wave, that's coming into our eyes changes as the color shifts. You also have amplitude, shown here in the middle row in the diagram on the bottom right. And amplitude is the height of those peaks. So it's the distance between peak and trough. It's how tall the wave is, so to speak. And that's what we perceive as brightness. If the difference between peak and trough is really small, well, that's not a very intense stimulus. If it's really big, well, that is an intense stimulus. We perceive it as being bright. So there's the amplitude, it's brightness, and there's the wavelength, it's color. And then you can also have a mixture of waves. We seldom have a color that's just one wavelength of light. We often have complex colors that are a mixture of wavelengths. And this is what we perceive as saturation. If you've taken a class in visual art or psychology of perception, or a computer science class in which you deal with images. Saturation is a little more abstract concept, but it's about the number of different wavelengths that are built into a stimulus. So it's about the purity of the stimulus. And these properties of light are picked up by what are called photoreceptor cells. And this is one of the first stages in our sensory process. So we have these cells that are responsive to light. So when light strikes these cells, they transduce it. They turn it from light into these neural signals. And we're not really going to go into the details of how that works, but there are molecules in these cells that change effectively neural activity when they're hit by light. So we talked about action potentials, and action potentials depend on the change in charge. And that's what photoreceptor cells do, is they end up changing charge, the charge inside the cell, as a function of how much light is hitting. So it transduces light into neural signals. And these cells are, of course, found in the retina, which is the part of our eye that actually responds to light. So the whole eye is not light sensitive. It's the retina at the back of the eye that detects light and converts it into these neural signals. Okay, so now that we understand what light is, it brings us to the eye. And when it comes to the eye, of course, we refer to the eye or the eyeball, the whole organ. Um, so we're gonna get to the retina, which is really the most important part of the eye for our purposes. So light eventually is going to hit the retina, which is what senses light. But to get there, it has to go through a couple parts of the eye. So it has to go through the cornea, which is the outermost covering of the eye. If you've ever gotten a scratch on your cornea, that is that outermost layer, which helps protect the eye. You also have the pupil, which is something we've all heard of. It's the hole, really, in the eye. It's that dark spot. And it's that dark spot which lets light into the eye. It's going to let light through and let it hit the retina. And then there's also the iris. The iris is most famous for giving people eye color. So when you say someone has blue eyes, for example, or someone has brown eyes, you're referring to the color of the iris. The outer part of the eye, of course, generally is white or close to it. Um, the iris gives us eye color, but more importantly for us, it also determines the size of the pupil. The pupil can dilate and constrict, and that is caused by relaxation and contraction of the iris, and that's what determines how much light gets in to hit the retina. In a bright environment, you don't need as much light getting in, proportionally, and so your pupils constrict. In a really dark environment, you want to let in as much light as possible so they dilate, they get bigger. Once it goes through the pupil, light then hits the lens. And this is not a glass lens in the usual sense of lenses. It's made of proteins, but it is a clear material. And importantly for us, it stretches, it's pliable. And so when you focus at different distances for a camera, what you do is you twist the lens, uh, the framework for the lens. And what that does is adjust the distance between the lens and the film or the digital detector. We don't have that option in our eyes. The lens does not move back and forth the way it does in a camera. Instead, what it does is it gets contracted or it gets relaxed. So the lens is not spherical, but it's sort of oval shaped, and it can be thinned out or let it get, or the muscles attached to it will let it get thick. And that's what allows us to focus at different distances. Now, as you age, your lens isn't quite as elastic as it used to be. And so that's why when people get older, they have trouble focusing, in particular on near objects. You often have to get reading glasses. And it's because your lens isn't as pliable as it used to be. But all that eventually lets light through, and it lets that light through to hit the retina, which is the part we care the most about in our topic today. And the retina is an array of cells. There's lots of different kinds of cells in the retina. We're not going to really talk about all of them. But the important job of the retina is to convert light into neural signals, that transduction. And so the retina has certain properties that allow it to function the way that it does. It sits at the back of the eye. It is what light hits after it passes through the lens. Incidentally, after it goes through the lens, the image is technically backwards and upside down. Uh, that's how it is on our retina. Of course, we don't perceive the world that way because as our visual system develops, we learn to not view the world as upside down and backwards. We invert the image, and we do that automatically. We don't think about it. We don't have to. But the retina has light hitting it, and technically the image is backwards and upside down. And the retina has two important kinds of cells for today. One are rods, and rods detect light. They're a type of photoreceptor cell, which we were just talking about. And in particular, rods work in low light levels. So when it's dark, particularly at night, you're mainly depending on your rods because there isn't much light around. And rods are more sensitive than the other type of cell we'll get to. However, they don't really detect different colors. All they can detect is overall intensity. So if you depend only on your rods, you see everything in grayscale. You would be colorblind. The other type of photoreceptor cell are cones. And we've probably heard of rods and cones. And cones complement rods. So they work best in higher light level conditions. So it has to be kind of bright for cones to start working. So they're not as sensitive as rods, but they come in a variety of types. And that's what gives us our color vision. There are different kinds of cones. And so depending on what kind of cone the light is hitting, that cone will become more or less active depending on what color that cone is tuned to. And so we have the diagram down here at the bottom left, where we see these different curves. And these curves represent the sensitivity of the different kinds of cones. So on the far left, that cone is sensitive to blue light. It'll respond somewhat to white light, for example, or green light, but it's really active for blue. The far right curve represents cones that are sensitive to red light. So if the light is red, it's very active. If it's green, it's kind of active. If it's blue, it's not very active at all. And so depending on the intensity of signals coming from the different kinds of cones, your visual system can deduce what color of light is being witnessed. So cones require high light levels, and they detect colors. And if you are colorblind, odds are you're not totally colorblind, but if you have a color vision deficiency, odds are it's because you have a deficiency in one of your cone types. So you can detect some colors, but not all the colors that someone with normal color vision can detect. Now, looking at this diagram in the bottom right, this is a diagram of the retina. And we see what's called the fovea, which is near the center of your vision, and it is the part of the retina that has the densest concentration of cones. There are almost no rods in it, and so it has cones packed together very tightly. And that is where you have your most accurate vision, and it's where you have your best color vision. However, it is not your most sensitive vision in terms of light level. So, for example, astronomers that are looking for very dim stars know not to look at the star with the center of their vision. They have to look a little bit to the side, because if they look straight ahead, that light will fall on the fovea, where all the cones are. And the cones aren't sensitive enough to pick that up. They look a little to the side. Now their rods can detect those dim objects. So the fovea has lots of cones. It's good for high-resolution vision. It's good for color vision. It's not good for dim conditions. And you also have, of course, the optic nerve, which is what exits the back of the eye and eventually goes to the brain. However, that optic nerve has to leave the eye somewhere, and it's a whole bunch of axons packed together. So the place where it leaves the eye is often called the blind spot, because wherever it leaves the eye, any light falling on that part of the retina doesn't get detected. There's all these axons passing out of the eye. There's no room for rods and cones. So you have no photoreceptors where the optic nerve exits, and so you get a blind spot. Now, of course, we're not aware of a dark field in our vision. We're not aware of a spot in our visual field that we can't see anything in. Our visual system actually fills in that gap. And your book has an example of an illusion that demonstrates this concept. But there are lots of these kinds of pictures you can look at. For example, if there's a gap in a bar at a certain spot, in a certain distance, that picture of the bar will appear continuous because your visual system can't tell what's there on the blind spot and infers from local information what's most likely there. So again, people have constructed illusions uh, that manipulate this aspect of the visual system to where you get a false inference about what's there in that blind spot of your visual field. Okay, so once light gets into the eye, hits the retina converts that light, depending on its color, intensity, and position, converts that into action potentials, into neural signals that then get passed on to the brain. So when it comes to getting from the retina to the brain, it has to go a fair distance. And so we're not going to talk about all the steps in detail. What's interesting is that vision goes not directly to visual cortex. It first goes to an area known as the lateral geniculate nucleus, sometimes just called the LGN. And this is part of the thalamus. As we'll see, the thalamus is the sensory routing center. All of your senses, all of your sensory information, except for your sense of smell, go through the thalamus. And vision is no different. So information from both eyes gets sent to the lateral geniculate nucleus, and then gets passed on to what is called primary visual cortex, often called V1. V for visual, 1 because it's primary. So V1, primary visual cortex, same thing. And that's where a lot of the most basic processing gets done. Your retina does a little bit, but your primary visual cortex will process things like shapes, the very elemental parts of shapes, lines really, lines of a particular orientation. And so these are very simple aspects of a stimulus. And part of what the visual system does is build up from these simple constructs like lines oriented in a particular position to things like shapes, eventually to things like objects, maybe even familiar objects. Maybe it's a face that we know. And so this hierarchy starts at primary visual cortex. One of the things that's interesting about visual cortex is that it's organized by what we call visual field. So, as we'll see later, your sense of touch is organized by the side of the body. So the left side of the body is processed by the right side of the brain. So it switches sides. But it's not the case that visual cortex, that left visual cortex processes the right eye and vice versa. Left visual cortex processes the right visual field. So if something is in your right visual field, some of that information will go to your left eye, some will go to your right eye. And what's interesting is that the nervous system sort of remixes information coming from the two eyes so that everything on the right half of your visual field gets processed by the left part of the brain, and everything on the left half of your visual field gets processed by the right part of the brain. And so you do have this, what we call contralateral organization, or this switching of sides, but it's not according to eye, it's according to which part of your visual world the information comes from. Now, once it gets to primary visual cortex, after V1 does some processing, it then gets sent to other areas that do additional visual processing. And your vision system can really be broken down into two pathways. That is, two sequences of regions that your visual environment gets passed through. There's what's called the ventral pathway, sometimes called the what pathway. And what the ventral pathway does is process things that have to do with the identity of the stimulus. Things like shape, things like the identity of the object. If it's a familiar object like a face or some other object that we work with a lot, we're obviously highly attuned to small differences in things like faces. We can tell when someone is our friend or our family member, that we've interacted with them a lot, and so we recognize them from their face. That requires a lot of detailed processing. And it's the ventral pathway that does that. It processes identity of objects, recognizes objects. In contrast, the dorsal pathway, so ventral and dorsal, if we recall, have ventral is the lower part of the brain, so the part toward the bottom. Dorsal is the part toward the top. Um, the dorsal pathway, sometimes called the where pathway, and it processes things like where a stimulus is in visual space. Not what it is, but where it is. If it's moving, the dorsal pathway processes where it's moving to and from, how fast it's moving. And these are different traits of a visual stimulus, but they all connect back to that stimulus itself. So stimulus, we have to know what it is, but we also need to know where it is and what it's doing. So these are the two pathways of the visual system, it seems. And this is a theme that does occur in other senses as well. We'll talk about it mostly here. Uh, but we have these two pathways, the ventral, which runs along the temporal lobe, and the dorsal, which runs from the occipital lobe up along the parietal lobe. All right, so we have our visual system. We have information going into the retina, going to the brain, being processed by different parts of our visual system. How do we get from that to mental representations of objects, which is really how we experience the world? Again, we don't look at the world in terms of collections of lines or even collections of simple shapes. We think of the world in terms of objects. And if those objects are people, then we think of them as people. And people have a special meaning. Organisms are treated in general differently than inanimate objects. So we have different categories for objects. And what this means is that the objects are defined by a lot of different characteristics. We were just talking about the ventral and dorsal pathways. Uh, and so objects can be defined by their shape, by their color, but again, we also classify things in terms of location, in terms of movement, uh, and that's just visual properties. Objects can, of course, have a lot of different aspects to them visually, and those different aspects are processed by these different parts of your visual system. Um, you can expand beyond that, and maybe an object is emitting a sound, or maybe the object has a particular odor. And so you have your visual system processing the object, then you have some other totally different sensory system processing the object as well. So how do we connect those different aspects of the stimulus? Well, that's what's called the binding problem. And it's called that because binding means to combine, to stick two things together. And so our brain has to have some way of taking an object and tying that object, that mental representation, to its visual properties, its shape, its location, but also to its properties from other sensory systems. Is it making a sound? Is it emitting an odor of some sort? Uh, and so all of those different properties, even if they're from different senses, have to all get associated with the object. And this is what, in particular, parietal cortex does. You have other association areas as well. But one of the things your brain has to do is reconcile the fact that all these different properties of the object are still associated with the object itself. That's not the only hard part about objects, though. You also have what's called perceptual constancy, which is pretty remarkable. So we have the ability to recognize objects even when many of their properties change. So your book uses the example of the actor Johnny Depp. So here we have a picture of him uh, in an everyday setting. And here we have him playing a role in a movie. And despite the fact that the lighting is different, his clothes are different, hair is different, complexion is different, uh, we're still able to recognize the person Johnny Depp in both of these cases. And we know it's the same object, so to speak, insofar as people are objects. Uh, and so the identity of the object obviously isn't tied to a particular appearance. Another example would be a chair. So if you're familiar with the chair and you walk around it and see it from a different perspective, you're able to recognize it as the same chair. just because it looks a little bit different by virtue of your having moved, that doesn't make it register as a separate object. You're able to alter the visual properties of an object, and it still counts as itself in your visual system, which again is pretty remarkable that all these different properties can change, and yet the object is still recognized as having this constant identity. So we still perceive it as being the same object. At a more basic level, the question is, what makes an object? Why, when is something an object? How does our visual system classify something as a single object? And there are a variety of principles that psychologists have come up with. One is simplicity. So an object is a unitary entity, uh, if that is the simplest explanation for the visual stimulus. So here, for example, you could identify it as a triangle sitting on top of a rectangle, or it could be a triangle sitting on top of a trapezoid, sitting on top of two rectangles. But none of those would be as simple an explanation as an arrow. So we have a single description for this object, as opposed to having multiple objects comprise it. Another criterion is closure. So our visual system is very good at filling in gaps. I've already talked about the blind spot in the retina, and that's at a very low level that gap gets filled in. But even for a complex visual scene, here in this diagram, we still see an arrow, even though technically it's a collection of 4 lines. Um, and in this case, our visual system is filling in those gaps. And we have to fill in gaps all the time in the real world. So if an object is partially covered up by something, then we're only seeing parts of the object, but we're still able to recognize it as a single whole. We still see the object for what it is. It doesn't become 2 objects just because something is obscuring part of it. And so this is an example of our visual system filling in the gaps so that we have that simple description of a single object. There's also the concept of continuity. So when we see edges, contours, our visual system extrapolates those edges to where they seem continuous. So in this diagram here, we could see this as 2 V-shapes. We could see it as 4 separate lines. But it's a much simpler explanation to see it as 2 long lines intersecting one another. So we can recognize that these lines, these contours, continue on even though they cross. And so that continuity is something else our visual system does in order to build simple visual constructs into objects. There's also similarity. So your visual system will group similar objects if they're the same shape, for example. They tend to be construed as a single object. Here in this diagram, you probably perceive it as 3 columns, as opposed to 9 individual shapes. Technically, it is 9 individual shapes, and you can force yourself to see it that way. But your visual system automatically notices the similarity between the triangles and the circles. There's also proximity. Objects that are close together, or properties that are close together spatially, tend to be grouped into single objects. You have a group of these things over here and a group of these things over here, as opposed to combining distant properties into objects. So proximity helps the visual system define objects. So they have to be simple, they should be as few shapes as possible, and they tend to be grouped together in terms of their properties. And then there's what's called common fate, which is sort of an odd term, but all that really means is if different aspects of an object, different parts of an object are moving in the same direction at the same speed, we tend to group them together into a single object. So in this diagram here, we have a road sign. So we've all seen those flashing arrows that move, that seem to move from left to right. However, of course, what's really happening is that one set of light bulbs is being turned on as another one is being turned off. The lights are not moving, and there really isn't an arrow that's moving. But because of the sequence in which the light bulbs are being turned on and off, we perceive it as a moving arrow, as opposed to a series of light bulbs that are being turned on and off. And things like signs in casinos and movie theaters use this principle also. We've all seen those borders of light bulbs. That as they turn on and off, it looks like a moving border. when in fact it's just light bulbs being turned on and off. But that's the idea of common fate. We tend to attribute the property of being an object to things if they move together. Okay, so we have some rules for what constitutes an object. We're able to handle a wide array of changes in properties, and we're able to combine those properties such that they still constitute a single object, as opposed to having all these different parts of our brain processing the environment, but not really communicating with one another. They talk to one another, and they're able to inform the single concept of an object. And of course, our three-dimensional world is even more complicated than that. We don't just have one object at a time, and those objects are often not that simple. So we have to process a visual scene and understand all the objects in it, understand how to navigate a three-dimensional world like that. And of course, one of the things we have to do with objects is determine how far away they are and how big they are. So we have to detect their depth and their size. Now, some of this you can do with just one eye. So we have two eyes, and that second eye does confer an advantage. It's not just a backup. But there are what are called monocular depth cues. And these are clues in a visual scene that only require one eye, but that help us say something about that scene, that help us describe what the objects in that scene are doing, or how they relate to one another, where they are. One rule is called familiar size. And this is exactly what the name sounds like. That's where an object that we already know, we can use as sort of a benchmark in terms of its size. We know how big the object should be, so depending on how big it looks, we can say something about how far away it is. So we know how big a quarter is, for example. So if it's a really big image of a quarter, we know it must be close, because quarters don't change size. If it's a really tiny image of a quarter that's hitting our retina, we know it must be far away, because quarters have a constant size. Here, this picture on the right is what's called the Ames room. And what's really interesting about it is that it is a room designed to fool your visual system. So you look at the room through a peephole from a particular perspective. And the thing is, the room is not composed of right angles. It looks like it is because it's been constructed and painted to look that way. But in fact, you see here in the right part of the diagram, you have two people that are of average size. One looks much bigger. And this is a confusing image because of familiar size. We know how big the average person is. And so you would think that these two different people are different distances. In fact, they are. But because of the room is constructed and the angle from which we're viewing it, we can't tell that the room is at odd angles. We can't tell that these people are actually quite different distances. So it gives us the illusion these people are very different sizes, which contrasts with our familiar size concept. We know how big these people should be on average. And so that's one of the cues that we use to say that something is odd about the Ames room. But in the average everyday experience, it also tells us how far away objects are. We know how big cars are. We know how big people are. We know how big houses are. And so we can use those benchmarks to say how far away things are. There's also what's called linear perspective, and this is illustrated quite well here by these railroad tracks. As parallel lines go away from us, the further away they are, the closer they appear. They converge on the horizon. And this is commonly used in things like visual art. But it happens in everyday life as well. You can see the lines on a road. As they go away from you, they start to converge. And so you can tell that something's going off into the distance because of this fact of linear perspective. You also have what's called interposition, which is just a way of saying that when you have two objects, if one is behind the other one, you can't see all of that farther object. So the closer object obscures parts of the farther object. And we can use this to tell which of two objects is closer. We can't tell how big they are necessarily, but we can tell that one is in front of the other, and therefore one must be closer to us than the other. So that's a few of the monocular depth cues. I'm not going to go through all of them. But you also have cues that require two eyes. And the most important of these is binocular disparity. That's a fancy term with a simple meaning. All that really means is that because your eyes are spaced slightly apart, they get a slightly different view of the visual world. So your right eye might see more of a surface, might see an edge that your left eye can't because your right eye is shifted to the side from your left. And so the fact that your two eyes will get slightly different images is what gives you a lot of your depth perception, your ability to tell how far away things are. And so that depends on binocular disparity. Disparity means difference between things. And so your two eyes get slightly different views of the world. And you can use that to say something about how far away things are. And of course, navigating your 3D world requires some attention. And attention can influence what we perceive. There is what is called change blindness. So depending on what we're paying attention to, parts of our visual world can change, and we may not notice it because we're not paying attention to them. And in particular, what change blindness is if the change is particularly slow, then we don't notice it. And there are lots of illusions that make this point. You'll have some picture, and very, very slowly, one object in the picture will disappear or appear. And because it's happening so slowly, your visual system accommodates those very, very small changes. And so you don't get that stark contrast. If you just show the beginning and end points of the image, it's very obvious. But because the change happens gradually, you don't see it. This has also been illustrated in a famous experiment where someone asks for directions on a college campus. And the person being helpful starts giving directions, and then a door passes between the two people, and the experimenter has switched out the person asking directions to a totally different person. And people often say they didn't notice that the identity of the person changed, even though they don't look at all the same. So those sorts of changes can be either gradual or they cannot be noticed because the person's not paying attention. And this leads us into what's called inattentional blindness. So not only are we not always perceiving changes, if we're not strictly speaking paying attention to them, also our attention can be focused on something else. So change blindness doesn't require focused attention. It just requires inattention to some aspect of the visual world. Inattentional blindness means you're paying attention to something, you're really concentrating, but you don't notice what would really be obvious to the casual observer. So there's a famous experiment shown here in the bottom right where participants watch a video and they're given a difficult task. They have to watch a team of people pass a basketball around and they're asked to count the number of times the basketball gets passed. This is not an easy task to do. People are moving around, the basketball's moving around a lot, so it requires a lot of attention. And what a surprising number of participants don't notice because they're doing this task is that someone in a gorilla suit passes into the scene, beats their chest, and then passes out of the scene. Now, if you're just watching the video and not told what to do, it's very obvious. However, if you are paying really strict attention to the task, then it's not so obvious, and you tend to miss these really glaring features of the video. Okay, that will do it for our unit on the senses in general and our visual system. Next time we're going to look at some of our other senses. So for example, we'll ask, how does the sense of hearing work? How do we get from what are effectively waves of air pressure in the environment to a perception of sound, and especially complex sounds like music or speech? We'll then move on to the sense of touch. And it turns out that our sense of touch actually combines a lot of different sense organs. We tend to think of it as a single sense, but it is in fact many. And then we'll get to our sense of taste and smell. And these both have a common element because they both detect chemicals. Taste detects chemicals in the mouth, smell detects chemicals in the air, but they're still detecting the presence of chemicals, which is very different from measuring light or measuring sound waves. So that will be the next lecture, and I will see you all then.